for RNA-seq, you prepared messenger RNA, and that's what you sequenced. Well, you converted it into copy DNA in the end, but you sequenced the messenger RNA. Now, as you know, RNA easily degrades. So there are machines that allow you to check degradation, but most labs do not have one. Your sequence provider should do it, but they don't always do it. So if you haven't checked the degradation of your RNA itself, then this is a plot that will allow you to check it now, after the mapping. So what does he do? He will take the BET file with all the annotations. So what does the BET file contain? All the transcripts. So he will take all the transcripts and he will divide all the transcripts into 100 equal parts. Yeah, so the first 1% of the transcript, this is the last 1% of the transcript. And he will do that for each transcript. So for large transcripts, these percentages are larger, these regions are larger. For small transcripts, they are relatively small. Then he will take the BUM file. Yeah? And he will see for each part, each of these 100 parts, how many reads were mapped there. And he does this for every transcript. So you can imagine this goes slow. Eh? Makes sense. And then he plots it. So what do you see on the x-axis? You see from 0 to 100. Yeah? So the 100 equal parts for all the transcripts. What do you see on the y-axis? The coverage. How many reads were mapped there? So how does it work now? Well, when you make your RNA-seq library and you convert your messenger RNA into cDNA, you typically use either oligo-DT primers, like you normally do in the lab, or you use random primers. When you use oligo-DT primers, it means that you're going to start your copy DNA formation at the poly A tail, because that is where the oligo-DT primer will bind, at the poly A tail. So reverse transcription will start at the poly A tail, at the 3' end of your transcripts. And most of the fragments that you will generate will be fragments on the 3' end of your transcripts. So when you have created your library like this, you will get a plot like the one that you see here with the not so bright colors, because all your fragments came from there. So you had, most of the reads will be representing this part of the transcripts. In that case, the plot doesn't tell you anything. If you made your library with random primers, you will get fragments from everywhere. In the transcripts, you will get fragments from the 5' end, you will get fragments from the 3' end, and you will get fragments in the middle. Okay. So for these applications, this plot is interesting, because what you expect there, if there was no degradation, you expect to see the bright plot. A high coverage over the entire body of the transcripts. If your RNA was degraded and you used the random primers, what will happen? Well, degradation of RNA always starts here at the 5' end, because the poly A tail shields the 3' end from degradation. So if you have degradation and you used random primers, you will see this. So the same thing that you see when you use oligo-DT primers. Low coverage at the 5' end, because this was degraded, and high coverage at the 3' end, because this is the last part that degrades. If you see degradation, is this bad? It can be, it depends. If you see degradation and you're only interested in differential expression, then it's still okay. You will have reads here, and you can use these. If you're interested in splicing, yeah, then this is really bad. Eh? For splicing, you need to have reads all over the entire length of your transcripts.